Okay, so we're going to get started. Thank you all for coming for the PhD defense of William Gilpin. He's a graduate student in applied physics. William is tough to introduce. I'll start with that. He came in a couple of years ago. He somehow figured out how not to change, although you can see some changes in William, but he's been forever young in spirit. When you see him in formal settings like this, he will look like one kind of a person. But I've worked with him for many years now, and this is another William Gilpin I'd like to introduce to you. And he's very driven. It's no matter what and where we are, he finds a way to come up with creative solutions to problems that he cares about. This is William and I in a swamp that will remain unnamed, trying to find carpenter diatoms while it's actually raining, which is not visible in this shot. This is another William out there sharing his passion for science with a lot of people. And I think both his formal and informal ways of science have really informed me in how to think about questions. Something that you will meet today, I wonder if we brought Mr. Pateria and Mrs. Miniata to the collection today. You're missing out. I was really hoping the actual contributors for this talk, the mother and father of the baby starfish that you'll hear about, are fossils in the lab now, but you'll hear the story of their offsprings, and of course, we were doing the experiments, and I learned that William had figured out how to keep these things forever in the lab is because of his incredible fossil collections that if you get a chance at some point of time, you should talk to him about. I think one of the remarkable aspects, in all seriousness, of a student like William is he's really an independent thinker. So I'm just so glad he joined the lab and we got to do some things together. He definitely moonlighted with Mark from time to time, and that led to a really incredible series of papers that he won't get a chance to talk about. But on holidays, he would go home and then come back with a paper that he independently thought and wrote. This was a title that first time I learned about this work was in Stanford News. Stanford physicists figured out how swirling liquids work similarly to bitcoins. Maybe while watching his cup of coffee at a winter break in Florida, he figured out a way to connect that to cryptography. So the next time you're swirling your coffee, possibly if it was up to William, you might mine some bitcoins. And with that note, I'm going to pass to William.
All right, thank you for the introduction, and thank you so much to everyone for coming out today. When I sent out this abstract talking about baby starfish swimming, I was sort of expecting to have maybe like my 5 committee members and maybe my girlfriend show up. So I really appreciate everyone for coming out this morning and hearing about this. But I should warn you that I actually won't just be talking about baby starfish today. I thought that would be a little bit more true to my trajectory during my time here at Stanford. And before launching into this specific project, I spent a little bit of time talking about a couple of projects I've worked on over the years. And this is more to give an overview of some of the different things I'm interested in and the sort of way I see things fitting together. So my plan today is that I'll first give an overview of some of the general areas I've had the chance to work on over the years. Then I'm going to talk very briefly about this Bitcoin, or we'll say cryptography project that Manu just mentioned. So I'll talk briefly about that, and then I'll spend, let's say, half an hour or so talking about this main Starfish project. So we'll start with a little bit more of an overview. Okay, so I'd say that broadly speaking, I've worked on 3 main areas during my time here at Stanford. So my official research, my official dissertation research as part of Manu's group, has been primarily at the intersection of biomechanics and topics in fluid mechanics. And we're very, very interested, generally speaking, in systems where we hope to be able to use physical forces and biophysical arguments to place constraints on the sorts of evolutionary strategies seen at the base of the animal tree of life. And the specific case I'll talk about today is the Starfish project, so you'll hear a lot more about this in a little bit. So separate from my work in Manu's group, as Manu mentioned, I've moonlighted almost my entire time here at Stanford as a member of Mark Feldman's group. And with Mark, I've worked on a variety of topics at the intersection of ecology and evolutionary theory. So there's a lot of different things in that domain, but one more theoretical problem that we've worked on recently has involved trying to take a classical ecological model, just a predator-prey interaction, and it's a well-known model because it shows continuous cycling in phase space, and we modified those equations to include the leading order effect of evolutionary interactions. And it turns out that when you add just this one additional term into the dynamics, you end up seeing a lot richer dynamical space. You actually see chaotic cycling appear in the predator-prey coevolution dynamics. So it's a more theoretical result. Also with Mark's group, I've also worked on a more applied set of problems, including a long-running collaboration with a group of anthropologists based in Tokyo, with whom we've tried to work on applying some problems from ecology and evolutionary theory to the problem of understanding why the Neanderthals suddenly disappeared 40,000 years ago. So it's a little bit more of an application of some of the theoretical work with Mark's group. So as Manu alluded to, separate from my work with Mark and my work with Manu, I have a few interests of my own, which are primarily the intersection of nonlinear dynamics, fluid dynamics, and problems in computer science theory. So there's the project I'll talk about next, which is this problem showing that you can build a cryptographic hash function using a fluid flow. And then very recently I've gotten more interested in seeing whether one can use tools from neural networks theory to pull out efficient low-dimensional representations of operators for dynamical systems. And I just wrote my first paper using this to study cellular automata. So this hopefully gives some idea of some of the different things I'm interested in. But I say that the common thread here is that even though these are very different applications, the tools and often the software and other things that have been developed in support of these projects have often had a lot of crosstalk across these different projects. For example, developing a Lyapunov exponent code for one project allowed me to make progress on another project. So there's been a lot of crosstalk even though these are three very different areas. Okay, so with that in mind, I want to launch into the first part of my talk, which is talking about this cryptography project. And the spirit of this project is trying to see if it's possible to generate and to represent ideas common to computer science using concepts borrowed from what's more familiar to me, which is the physics of fluids. Okay, so what is a hash function? So generically speaking, in computer science, a hash function is any function that maps some high-dimensional input to some fixed-dimension output. So in a digital context, this would be something that can take some arbitrary sequence of ones and zeros, this arbitrary binary string, and convert it into a fixed length binary string. In this diagram, I said 3 bits. And so the whole idea behind a hash function is that regardless of the input, it'll always give you an output of the same length. And so for that reason, hash functions are very useful in contexts where I might want to be able to represent a piece of information without storing the full piece of information. So one can imagine something like a database system where I just check that I have a hash of something rather than storing all pieces of data. One of the main applications of hash functions is that they appear a lot in cryptography contexts. So this includes things like Bitcoin, but also includes more sort of standard things like email signature verification algorithms. And the idea here is that hash functions have this ability to map an arbitrary input to a fixed output. And so if I have something like a password, instead of having to store it as a list of user passwords, I can just store the hashes of those passwords. And so then when someone types something into their password box, I can just check that the hash is correct rather than having to have an original copy of the password. So the whole idea behind a hash function is it allows you to have a short fixed length representation of a piece of information. And in order to be useful for a cryptographic context, these functions have to satisfy certain properties. They have to be non-invertible, that way I can't just guess the password from the hash. And importantly for today, these hash functions have to be very, very sensitive to the inputs that they're given. So if I give one of these hash functions some long, arbitrary string of ones and zeros, and I then mutate one bit in that string, I should be able to run that through the function and get a very different hash. So my function should be able to differentiate small changes in the input that it's given. So a few years ago, I was thinking about these hash functions and this idea of being very, very sensitive to the input that you're given, and it reminded me generically of things that pop up a lot in dynamical systems. So the video here is just a very typical video of a flow near a hyperbolic stagnation point, just a 2D flow. And you can see in this video, two particles start out very, very close together, and then just due to the dynamics of being subject to the flow, they drift apart over time. And so this idea of starting out very close together, but just under the natural action of the map ending up in very different places, this to me sounded like it had some analogy with this idea of being very, very sensitive to the input. So I wanted to see if there was a way to take this sort of loose analogy and try to formalize it in a little bit more detail. So in order to make this a little bit more precise, I need to use slightly heavier machinery than just something like a stagnation point. Because you can imagine that in this system, the reason that those particles drifted apart over time is because I happened to pick them so that they straddled a separate matrix. Right, they're on opposite sides of that stagnation point. But if I picked any other pair of points in the domain, those points wouldn't necessarily have this separation property. So the general purpose solution to get around that is to use a class of dynamical systems called chaotic maps, which are basically just dynamical systems that pull apart most trajectories over time. So any randomly chosen pair of points in the domain has a pretty good probability of being exponentially separated over time. So you can think of that stagnation point as sort of being blurred out over the domain, so that now it's a set that has a measure greater than just a single point. So this is just a general class of systems, and today we're going to talk about a very specific example of that class called the blinking vortex map. So this is an example of a fluid flow that has a chaotic property. And the flow itself is pretty straightforward, which it basically just consists of a closed circular domain containing a fluid. You can treat this as a 2D problem. And then the fluid contains two stir bars. So those stir bars, one is on the left side of the domain and one is on the right side of the domain. And the way that this flow evolves over time is that at t equals 1, I turn on the right stir bar. And then at t equals 2, I turn on the left stir bar. Then I turn on the right, then I turn on the left. So the flow is time varying because I alternate the left versus the right-hand sides of the flow being modulated by the stir bars. So it's a time varying flow, but it's time varying in a periodic way. So under the action of that time varying flow, the trajectories of advected particles have this property where they'll orbit around one stir bar. Then when I switch to the other stir bar, they stop, then they turn, and then they orbit around the other stir bar. So it has this sort of periodicity where particles orbit, then turn, then orbit, then turn, repeatedly. And it turns out that just due to the natural properties of this map, this has this property where most trajectories subject to this flow become strongly chaotic over just about 10 or so iterations of the map. And so the top row of panels in this figure just shows path lines with a handful of particles. But to get a sense of what the chaotic property means, this bottom set shows the results of applying this map to a very tight bar of initial conditions. This is just a tight collection of points. And you can see that collection of points starts to stretch out after the first few iterations of the map. And then after many iterations of the map, you can see it's blurred out into this complex attractive shape. And if you kept iterating this map, you'd eventually find that the probability of a given point being somewhere in the domain ends up becoming uniform. And so this happens to have this property where this entire set of initial conditions got spread apart over time, which is the really critical property of chaotic maps for something like a hash function. And a few more technical details about this map is that this map happens to have some nice properties for this application. It has a uniform distribution of the Acknowledgement exponents, and the size of the chaotic attractor happens to equal the domain. And so that's specific to this map. So generically speaking, this map is a well-behaved, well-defined chaotic map, and I want to see if it would be possible to use this map to generate a provably strong cryptographic hashing scheme. So the way that I'm going to use this map to do that is I'm going to take that idea of the flow evolving according to left, right, and I'm going to say that series of operations on the flow, that series of stirs, is something like a binary string. Right? So if I have left, right, you can think of that as just being 1, 0, 1, 0. So one can imagine then substituting that series of 1s and 0s with just some other binary string. And so that means that you now have a time-varying protocol describing the evolution of this flow that specifies some arbitrary piece of information that I might want to generate a hash of. So the input here is the time-varying sequence of parameters describing the evolution of this flow. And I should note that what I just described, this general scheme, it isn't necessarily specific to this map. One could imagine that if I had something like continuous time variation, I could use something like analog encryption or something to put arbitrary information into that flow. So the general scheme here is to just use time-varying parameters governing a flow to store information, and the specific application is this binary string that works with this blinking vortex map. So that's how the information gets into this system. So then knowing that, the way that we're going to generate hashes with this system is pretty straightforward, which is that I'm going to put a finite number of particles into the flow. So I've labeled those just with numbers in the top panel. And then I'm going to play the map, which is dictated according to the message, and then that map will just advect these particles over the domain. And so then after the map is played through the domain, I then have these particles in these pseudo-randomized positions within the domain, and I'm just going to read out the relative x coordinates and use that as the hash for my system. So I'm basically just using a permutation of particles subject to the map as the hash for my flow. So when you put all that together, the process of generating hash looks like this. So if you notice the particles, if you read their x coordinates, they just go 1, 2, 3, 4, 5 from left to right. So then I start playing a message here, and you can see that the particles, just as the flow evolves, they start to exchange positions. And so as they exchange positions, they end up being mixed together in such a way that after enough iterations of the map, the probability of observing any given permutation of these 5 particles ends up approaching unity, or it ends up approaching a uniform distribution. So the hash space for this is combinatoric, so it's going to be 5 factorial possible hashes here. So this is generally how the approach works. And so the question is, while this works in principle because it has this arbitrary length input going to a fixed length output, we don't necessarily know that this is going to satisfy the strongest required properties of a hash function. So what I did is I took this map, this blinking vortex map, I took this idea of being able to take arbitrary messages and write them into the flow, and I generated an ensemble of 30 million random messages, and then I looked at the properties of the hashes that those messages produce. And the result of doing that allows me to do statistical tests to make sure that these hashes satisfy the properties that would be expected of a good hash function. And the two that I really looked at is that I wanted to make sure that the messages themselves affected the final hash so that you had this sensitivity. And I found that as the message length or the relative vorticity, which is a measure of the Lyapunov exponent in the system, as both of those got large enough, the system approached the amount of decorrelation that would be expected for a theoretically optimal hash function. And then there's some other sort of finer statistical properties that hash functions have to satisfy. For example, 2 randomly chosen messages should have a certain probability of producing the same hash that's relatively low. And you can test that, and you can find that this system also approaches that performance limit in the limit of having a large enough Lyapunov exponent. So the system performs as well as can be theoretically expected from a good computational hash function in a digital context. So just, there's something to this idea of using this chaotic system as a way to generate hashes. So that sort of tells you how this system works, but I think there's actually a few reasons why I think there are some applications to this. Because this is sort of a proof of concept in itself, this idea that we can make this mapping between these 2 fields. The reason I think that this is interesting is because one of the things that popped out in this paper is that the ability of this function to generate hashes had to do with its relative ability to invert the positions of bits that comprise the hash. And it turns out that the reason that happens in this map is because this blinking vortex flow is part of a class of chaotic flows that has this quantity called topological entropy, which is basically a measure of how much the world lines of particles affected by the flow exchange and interpolate positions in irreducible ways over time. So the blinking vortex flow is a well-studied example of a map where the trajectories of particles actually braid together in irreducible ways across space and time. And so one thing that I'm seeing in follow-up work is that many maps that have similar quantities of topological entropy will have similar hashing properties. And that's because this idea of mixing trajectories is very closely related to the ability to mix together the different particle locations when generating the hash. So this is more of, I think, the conceptual contribution of this work, is that it shows that you can make this mapping between these sort of concepts that we use a lot in dynamical systems theory and the sorts of heuristics that might be important in a digital context. So a little bit more of I'd say an applied note, when I was making this system, one thing that I had in mind is whether it would be possible to use this idea of hashes in some sort of hydrodynamic context. And so one sketch I have of how this might be useful in a fluid dynamics context would be something like in a complex microfluidic device, where one could have some very specific series of operations being performed on some fluid as part of a microfluidic assay. And I might want to be able to have some low-dimensional way to verify that the steps in that process occurred correctly. And so I can envision this hashing property being used as some sort of microfluidic proof-of-work system to verify the steps of some complex process. Okay, so that's about what I wanted to say about this project for now. But I think that one of the big takeaways I want to emphasize about this project is that it shows that even this well-studied chaotic map, the blinking vortex flow has been known about for about 40 years, even this well-studied map can still have really surprising properties when we analyze it carefully. And that's something that's popped up a lot in fluids, is that even supposedly simple examples can still have a lot of dynamical richness. And so I want to change gears now and talk about a different project for my PhD that represents an example of a natural system that takes advantage of some of the capacity of fluids to do really surprising things. And so this next project was performed as part of Manu's lab in collaboration with a postdoc, Vivek. Okay. So a few years ago, I had the chance to take a class on marine embryology down at Stanford's Marine Station down in Monterey. And this was a hands-on class. We actually lived at the Marine Station for 3 weeks. And we would wake up in the morning, we'd go down to the tide pools there, and we would actually collect animals to take back to our classroom. And as part of the course, we would learn how to spawn these different marine animals, and then we would watch how their offspring developed over the course of the class. So it was a hands-on marine invertebrate development course. And so this course was very fun because it was a chance to see some of the diversity of different organisms that are in the ocean, and to see the different sorts of adaptations found among different invertebrates. But one of the surprising common themes that popped out of the course is that for a wide swath of different marine invertebrates, most of these invertebrates undergo a two-stage or biphasic life cycle. So most marine invertebrates actually start out life not looking at all like their adult body plan. So a good example of this is a starfish, which starts out looking like these little tiny swimming animals, and then after about 2 months, the starfish metamorphoses into its adult body plan. And similar two-stage developmental cycles are seen in everything from corals to sea urchins to a lot of other marine invertebrates. So this idea of metamorphosis is something that's very, very strongly conserved among a wide range of marine invertebrates. So I thought that this idea of having a two-stage life cycle was really interesting because one of the things I learned during the course is that the study of the invertebrate development cycle actually has a very long history in the biology community. So the idea here is that back in the late 1800s, when people were trying to build trees of life, they turned to looking at the comparative invertebrate development biology of different organisms as a way to try to piece together how different organisms might be related to one another. And the reason for that is because across a huge swath of different invertebrates, you can see a lot of diversity in the way that the larvae appear. So the panel on the right shows some of the diversity of marine invertebrate larval forms. And so people tried to very carefully compare and contrast these larval forms to try to get any sort of clue as to how different invertebrates might be related to each other. So there's this long history of people in the field trying to understand how invertebrates are related to each other and to try to piece together how these different larval forms might be related to one another. So this is what got me interested in these larvae as sort of this standing question that might have some interesting questions to remain uncovered. So during the course, one thing that struck me about these invertebrate larvae is that these invertebrate larvae are actually surprisingly good swimmers. So this is again a video of starfish larvae. These guys are about 2 weeks old. And they're swimming here just freely in a petri dish. And you can see there's actually a lot of complexity in how these animals move through the water. So you can see examples of these animals. They do these nice barrel rolls as they move across the field of view. Some of them do these really big loop-de-loops. And at one point in this video, you can see an example where two of the animals actually, they come up and they bump into each other, and then they do this very polite three-point turn in order to change direction. So these animals have a lot of complexity in the way that they've evolved to move through the water. So we were in the course, we were looking at these invertebrate larvae, and we were curious if there was something underlying the complexity of swimming, if there was some interesting fluid mechanics behind the complexity of the way these guys move through the water. So we did the simplest possible fluid dynamics characterization experiment that we could think of, which is that we took one of these larvae, we put it under a slide, and we seeded the water with neutrally buoyant particles, so that we could image the flows being created by one of these animals as it swims. And when we did that experiment, this is what we saw. So here we've taken a video of small particles that are neutrally buoyant that are being ejected by this animal as it swims. And then I've run this algorithm that basically just blurs the trajectories of particles across time, such that the length of a streak that you see is proportional to the velocity of the particle. And when we run that analysis, we can see these animals actually create up to a dozen vortices around their periphery as a function of time. And these vortices are actually very complex and very dynamic. So you can see examples in this video where opposite-handed vortices will actually coalesce, and then they'll decoalesce at later times. And so this animal is actually orchestrating the flow around it with a pretty high degree of sophistication. And you can see a lot of other really interesting phenomena. You can see stagnation points and other sorts of things in the flow. And we were really, really surprised when we saw that this animal was creating this complex flow field because this animal is a pretty solidly low Reynolds number swimmer. The Reynolds number in the swimming varies between 0.01 and 0.1. And so importantly, that means that at that length scale, creating vorticity is actually very, very energetically expensive. So these animals are continuously shedding power into the water to generate vorticity. So this is in contrast to other fluid dynamical systems that you may know that generate vorticity, things like fish swimming or birds flying, where generating vorticity at higher Reynolds number is generally less expensive. At this length scale, power dissipation goes as vorticity squared, and so this is actually a very, very expensive thing for the animal to be doing. So when we saw this, we were really, really surprised that something at this length scale would go through all this trouble of creating these patterns in the water because this is such an energetically expensive thing to do. So just to put this observation in context, there is a bit of an existing literature on people studying how organisms swim at low Reynolds number. But most of this existing literature on swimming at low Reynolds number, as far as we know about, has primarily focused on how single microorganisms swim. And so these are things like sperm swimming, or bacteria, or some types of algae. And so a lot of the existing literature on low Reynolds number swimming primarily focuses on organisms that just have one or two flagella that they're using to push or pull themselves through the water. And so a lot of the existing literature that we are reading about these low Reynolds number swimming would report there being basically two vortices in the swimmer frame due to the relative location of the swimming apparatuses relative to the center of drag of the organism. So we knew about this existing literature that talked about this 4-apt asymmetry, but when we saw that we had a dozen vortices, we thought that we really had found something interesting. So the reason that our system has so much more complexity in its flow field is actually, at least on the surface, pretty straightforward, which is that our system, instead of just having one or two flagella that's using to push itself through the water, the starfish larvae actually have up to 500,000 separate cilia on their surface, which are all beating at a given time. And these are very standard cilia. They're a little long, they're about 30 microns long, and they have a frequency of about 4 hertz. And these cilia are concentrated in these very dense structures called ciliary bands, which outline the periphery of the animal. So the animal, you can think of it as something like a sheet, and the edges of this sheet are where the cilia are primarily located. And the density of cilia within that sheet can reach as high as 10 cilia per linear micron. So these are very, very high density cilia. They're all packed together, beating jointly on the surface of the animal. So it makes sense on the surface why these animals might be able to access such complex flow fields, because they literally have many more degrees of freedom than most swimmers at low Reynolds number. So we knew that, so when we saw these animals had all these cilia and that they were creating these complex flow fields, it was pretty easy to get at least an intuitive picture of the origin of the flow fields that we're seeing, which is that the effect of the cilia is essentially to create a boundary condition on the surface of the larvae. So the animal itself is about, let's say, 600 microns to 1 millimeter. The cilia are 30 microns. So at the level of the flows, we can basically treat the cilia as creating a tangential boundary condition on the surface of the animal. So you can imagine that if all that these animals wanted to do was swim, then they would just tell their cilia to push water past their surface. That would be the boundary condition. The right side would push water this direction, and the left side would push water this direction. And so you just have streamlines that come in, go around the animal, and go out the other side, and you have this very uniform boundary condition on the surface of the animal. And so what we realized is that when we're seeing vortices around these animals, what we're actually seeing is the effect of portions of the ciliary band that are beating in reverse relative to this prevailing swimming current. And so these localized reversal regions on the surface of the animal are what give rise to these vortices that we see when we zoom out on our data. And the idea is that whenever a particle is coming in along a streamline and it encounters a reversal region, that forces its streamlines to lift off the surface and creates this localized eddy. So this idea of a boundary condition change is what's driving these complex flow field phenomena that we see when we zoom out on our data. So that made a lot of sense to us intuitively that basically this is a boundary conditions problem and that we just need to keep track of what the tangential boundary condition is. But one thing that we saw in our data that we weren't expecting to see is that when we actually zoom in on the boundary condition and we image the cilia, if we look at the sites where the boundary condition changes sign, we can actually see characteristic features in the ciliary array to support the change in the boundary condition. So we refer to these as topological defects because they correspond to a discontinuity in the local thrust vector associated with the cilia. And what's really interesting about these defects is they sort of match our intuition, which is that if you have a case where two streamlines are converging on the surface, there are cilia that get caught in the middle that don't know which direction to be. And so they actually end up getting tangled together. And then in the other case where you have streamlines that diverge on the surface, you see examples of the cilia splaying apart. So it matches what you would expect just by looking at the shear. So we saw these features and this sort of closed up our intuition, which is that suggested that the cilia are actually creating features to support this change in the boundary condition on the surface of the animal. So one thing, when we first saw these, we got really excited because we were hoping that maybe these defects were something like a dynamic feature and that we would see stuff like traveling waves of defects, et cetera, et cetera. But upon further investigation, it looks like these defects are actually something that is anatomically determined on the larvae. And the reason for that is because we always see these topological features in the same points on different animals, and we see examples where the defects actually break up and then reform in the same location sometime later. And so these defects appear to be something that the animal always puts in the same place. And from what's known about the larval nervous system in the nervous system of echinoderm larvae, it appears that the locations of these defects localize to separate regions of the ciliary band that are under the control of different parts of the larval nervous system. So the locations and placement of these defects likely has something to do with how the animal is coordinating and controlling its large-scale flow field. So at this point, we had a pretty good sense of how our system worked on an intuitive level. It's creating a boundary condition on the surface, on its surface, and that boundary condition cooperatively gives rise to the flow fields that we're observing. So we wanted to take what we'd seen and use it to put together a minimal model just to give an idea of what the different effects that we're seeing in the hydrodynamics actually look like. So our model is pretty straightforward, which is that we're going to ignore the geometry of the animal for a second, and we're just going to treat this like an arbitrary circular swimmer imposing a tangential boundary condition on its surface. So this is a very textbook type problem where you have a no-flux boundary condition, but an arbitrary slit boundary condition on the surface of a circle. And it turns out that this specific geometry is analytically solvable. It's a well-known solution to Stokes' equations called the Squirmer model. And this solution is actually very, very intuitive in a sense because it suggests that the flow field has this hierarchical form. It reminds me of something like a multipole expansion of electrostatics. And the solution, what it basically tells us is that the flow field we can think of as being organized in terms of these separate length scales associated with separate terms in the solution. And so that first order term in that solution, that B1 solution, that's the only term that survives as R goes to infinity. And so that term actually ends up being the term that sets the swimming speed of the animal. So that first order mode is the swimming speed. It's a system scale mode. So then all of these higher order terms, the B2, B3, et cetera, those correspond to adding progressively higher and higher order terms on that correspond to smaller and smaller length scales. And so each of these is going to be responsible for generating different numbers and different sizes of vortices. So as I add higher order terms, I get more vortices at smaller length scales. And so this gives us some intuition for how the flow field might be organized, which is that there's this hierarchy based off of how many reversal regions there are on the surface. And these B coefficients themselves are actually pretty straightforward to understand, which is that they're derived from the boundary condition itself. So in that case I mentioned before, where you just have this uniform boundary condition that pushes water past the surface, that corresponds to having no reversal regions. So the coefficient B1 just corresponds to the amplitude of that term. So if I had a solution where I just had no reversals at all on the surface, then all of my higher order terms would go away and I would just have the swimming speed and I would have no vortices. And so all of the reversal regions essentially serve to contribute to the higher order terms. And intuitively, the smaller scale of the actuation and the reversal on the surface, the higher order terms in the solution I'm going to activate. So there's this very natural hierarchy that pops out of this analytic model for the vortices being created by the animal. One thing to note is that the case I showed earlier that people had studied previously of a sperm swimming or an algae swimming corresponds to this solution taken to just the second order term. So to our knowledge, ours is the first low Reynolds number system that activates the higher order terms in the solution. So we thought that we were the first to see these higher order terms appear in the solution in an experimental system. Okay. So when we had this solution, we had a pretty good idea of what's going on, at least at the level of the flow field. But the obvious question is why these animals would even bother going through all this trouble to create these flow fields. What is the actual purpose of these flow fields? So one thing that we saw in our experiments is that these flow fields that these animals create are actually very, very good at entraining particles. So here I have a modification of the experiments I've been showing you guys, where this time I've put in slightly smaller tracer particles, about 0.5 microns, and I've added motile algae, which is the primary food source of the larvae, to the water. And you can see that the algae have this helical swimming trajectory, so when I do this motion blurring, they look like these little squiggles. And you can see that they wander around when they're far away from the animal, but when they get to one of these high vorticity regions near the surface, they tend to get entrained and caught up in those vortices. And that makes sense because the velocity of the fluid in those vortices can reach as high as the hundreds of microns per second, which is much faster than the 50 or so microns per second that these animals can swim. So they literally are being overpowered when they get entrained within these vortex regions. So when we saw that there was this entrainment happening, we became suspicious that what we're seeing really represents something like a feeding current being created by the animal. So when we saw that this was happening, we started looking at our experiments over slightly longer timescales. And one of the things that we started to observe is that these animals are actually continuously eating during the entire time that we're doing our experiments. So here I've just focused on the stomach of one of the animals. And you can see the animal just is continuously taking particles into its stomach over the course of, in this case, I think a 15-minute experiment. So these animals are feeding, and this video is sort of a special video because this animal actually overeats. So you can see it actually spits up some of the particles that we gave it. But at least some of the particles progress further into the digestive system. So these animals have a mechanism of getting particles out of these entrained vortices into their stomach. The actual mechanism that you go from being entrained to ending up in the stomach is actually known about for echinoderm larvae, which is that their cilia have this secondary function where they can actually lock down on particles that intersect with the cilia. So if you have a particle that's entrained in a vortex, if it gets close enough to actually touch a cilium, the animal has this response where the cilia can lock down on that particle, and then they have this mechanism of passing the particle along the ciliary band to the mouth. So there's basically two stages to the feeding mechanism in this system. There's this large-scale entrainment mechanism, and then there's this interception mechanism when particles get close enough to the surface to touch a cilium. And those two effects jointly contribute to what we observe as the feeding process. So knowing that this interception effect is present in our system, we can then return back to our original model and add in that interception to actually model the feeding as a function of the swimming stroke. So here what I've done is I've taken our model and I've basically plotted three different solutions corresponding to different numbers of vortices being created on the surface of the animal. I've then added this rule where I define a region proportional to one ciliary length around the animal, and particles that pass within that region have some probability per unit time of being captured out of the flow. So you can think of it as like the surface is sticky, and if particles pass close enough to pass within that region, they have some fixed probability per unit time of being captured. And so this has some form of something like a first passage time problem. And what we observe when we look at these simulations across different swimming strokes is that as we add more vortices into the system, we see an increase in the expected feeding rate. And this arises due to two effects. The first is that as you add vortices, you increase the literal hydrodynamic cross-section of the animal, so a greater fraction of particles will actually get close enough to the surface to get intercepted. So that's one effect. But the secondary effect comes from the fact that these animals actually have a low probability per unit time of capturing any one particle. And so by having these slow reversal regions near their surface, you have particles that slow down near the surface, thereby increasing their residence time and thus probability of being captured. So there's two effects contributing to the scaling of the feeding rate with the number of vortices. So one thing that pops out just very naturally of these simulations is that because there's only a finite number of cilia on the animal's surface, whenever you add to higher order modes, so you add more vortices around the animal, you always are doing so at the expense of that first order mode in the squirrel model. And the reason for that is because there's only so many cilia, so if you just wanted to swim, you just allocate all of your cilia towards that first order mode. And so whenever you're tiling in these patches that feed in reversal, those are always going to undermine your first order swimming mode. And so as a result, as you increase the feeding rate by adding vortices, you always observe a decrease in the corresponding swimming speed. So there's basically, at least in the space of the swimming strokes, there's this trade-off between the feeding rate and the swimming speed in this system. And this just pops out of the fact that there's a finite number of cilia to allocate. So when we saw that there was this trade-off between swimming and feeding in this system, we got really excited because it sort of gives us some sort of guide to understanding the different swimming strokes that we might be seeing in the system. But the nice thing about working on a micro swimmer that actually has a nervous system is that it's not always this simple as just classifying the swimming strokes, because the animal can actually change its mind regarding the flow field that it wants to create. So this is an example of an animal that actually can't make up its mind regarding the swimming stroke that it wants to create. And so it actually goes in this video between creating 4 vortices, to creating 6 vortices, to creating 2. It's continuously changing the flow field that it's creating. And this change in the flow field is corresponding to a change in the boundary conditions because this is a low enough Reynolds number system that the vortices are the result of boundary conditions. So what we're seeing here when we see the flow field changing is we're seeing a manifestation of the animal itself changing its boundary conditions. So we're seeing the animal's behavior projected into the space of the fluid's motion. So when we saw that there was time variation in these flow fields, we got really interested in seeing whether we could find a way to analyze the unsteadiness in the flow as something like a low-dimensional behavior problem. So we took these data sets showing time variation in the flow field, and we came up with a tool that allows us to take these flow fields, which are just essentially videos showing an unsteady flow. We have mechanisms of pulling a velocity field from these videos. We can take these long videos and then use them to generate these time graphs corresponding to the tangential boundary condition that matches the flow that we observe. The nice thing about having a low Reynolds number system is that the boundary conditions are equivalent to knowing the full flow field. And so this is really nice because it means that we can take these long videos of time variation in the flow field and we can just boil them down to these time graphs showing the tangential boundary condition as a function of time. And these time graphs are interesting because they show some of the time variation in the boundary conditions that the animal is creating. And they also actually show the defects, these sign changes, which correspond to those high frequency or high spatial frequency variations in the coloration in the chimograph. So this basically allows us to boil down our data to these nice chimographs of the corresponding boundary conditions. So once we have these boundary conditions extracted, we can then further boil them down by taking these boundary conditions and fitting them to the equivalent analytic model. So we can go back to the score model, which is parameterized by these B1, B2, B3, etc. coefficients, and we can take these boundary conditions and fit them to the model at each time point. So then we basically parameterize the behavior of the animal as a time series of the coefficients corresponding to the best fit score model at every time. And so these coefficients allow us to basically look at the animal's behavior in this space of a finite number of Fourier coefficients. We usually found that the first 5 to 10 were sufficient to describe most of the variation that we saw. And the one thing to note is that this is the first 5 components that I plotted here. And as I mentioned, only the first order component sets the swimming speed, and so that corresponds to that red trace. And you can see that the red trace, when it increases, it does so at the expense of the other modes. And so you can see the effect of this trade-off between allocating cilia towards the swimming speed versus these higher order terms. So as a check on this whole process of taking this data and fitting it into a model, another thing we can just do is take our original velocity data and run PCA, which is a dimensionality reduction technique. And when we run PCA on our data, we observe that there are basically 2 principal components that span this system. And they appear to be anti-correlated. If we look at the principal components themselves, one corresponds to what looks like a swimming flow with few vortices, and the other corresponds to what looks like a feeding flow with many vortices and a lower swimming speed. And you can see that by the fact that this first order principal component matches what the red trace is doing over time. So this is just a sanity check on the model that we're fitting to our data in case the geometry of something turns out to be important. So this is how we take our unsteady velocity boundary conditions and convert them to some estimate of what the animal's behavior might look like. So when we saw that, we wanted to take this data and look for patterns in the long-term behavior of the animal over time. And one of the things that started to pop out of our data is that we observed that the animal actually has a very clear periodicity in its behavioral variation over time. So we have this way of analyzing behavior, but we notice that just unprompted, the animal has this phenomenon where it will continuously be in one swimming mode, this first order PCA mode, for an extended period, and then it will transiently blink into another behavioral mode corresponding to having many vortices. And this is very consistent in timing and duration across multiple animals. So we saw that there was this rhythmic variation in the flow field, and this got us really interested in whether there's some sort of complexity to the way these animals have behavioral variation over long time scales. Because these animals basically do this blinking phenomenon independently of the external conditions. So there's this rhythm that appears in the way that the animal modulates the swimming stroke over time. So this blinking behavior is what we started calling it. It's something that has been observed before in a lot of other invertebrate larval systems. We saw it before in another system during the course. And there's a pretty large literature on invertebrate neuroanatomy that actually has studied these blinks in the ciliary beating conditions and which uses them as something like a readout for the effect of drug perturbations on the larval nervous system. So it's known that this blinking phenomenon is something that's observed across a lot of different invertebrates, and it's known to be related somehow to the larval nervous system. And so we got really interested in whether there's some hydrodynamic mechanism at play underlying this unsteadiness in the flow fields that these animals are creating. So in order to test that, we came up with a way to inhibit this blinking behavior using this known perturbation to these animals. If you soak these animals in magnesium chloride, it inhibits the ability of the animal to coordinate activity across its ciliary band, and it also paralyzes muscle actuations. And so we found that by soaking the animals in this drug can basically turn off all behavioral variation. So these are just 2 time lapses of particles under fluorescence around the animal. And you can see that the normal flow field is pretty complex looking. It has things like streamline crossings and whatnot due to time variation in the flow field that allows particles to make right turns and stuff like that. But when we treat the animals with the drug, all that complexity just goes away and we basically see a static flow field. So the drug doesn't disable the ciliary beating, it just disables changes in the ciliary beating. It locks the animal into one behavioral mode. And when we do our fancy chimograph analysis, we go from having all these high frequency components to basically just having radio silence over 15 minutes. So this drug allows us to very carefully check to see how this behavioral variation might be at play in the hydrodynamics. So under the action of this drug, one thing that we see is that the animal's overall feeding rate actually decreases quite a bit when we inhibit the behavioral variation. So in these 2 videos, the thing to look at is the number of new particles that arrive in the stomach, not what's already there. And you can see that the animal that has been treated with the drug has many fewer arrivals than the animal that is just untreated and is just being imaged continuously. So the feeding rate is affected by this drug. But that doesn't necessarily tell us anything, because it could just be that the animal's unhappy, it doesn't want to eat, etc., etc. And so as a check on that, what we can do is we can then take animals and we can just run particle tracking to see how many particles even get close enough to the ciliary band to get intercepted in the first place. And one of the things that we observe is that when we apply the drug to these animals, not only does the feeding rate decrease, but the number of particles that get close enough to the ciliary band to even get intercepted decreases. So the animal doesn't just eat less, it has fewer opportunities to eat due to the effect of inhibiting its behavioral variation, which made us start to wonder whether there might be a hydrodynamic effect here in the larval feeding rate. So in order to quantify that, we returned to these simulations we do of this simple model of a swimmer, and we just took that model and we just made these coefficients time varying. So we generalized the classic swimmer model just by having time varying coefficients. And we did this in the simplest way possible, which is that we said that there are going to be basically 2 timescale parameters that matter, which is the relative amount of time spent in the swimming mode and the relative amount of time spent in the feeding mode. So that gives us 2 parameters to search in a simulation space. And when we search that space of possible parameters, we see that there's this predicted optimum in the feeding rate at intermediate values of both parameters. So when we then take those timescales and we compare them to those that we measured in our experiments, we see that our experiments localized to around that peak, suggesting these animals are varying their behavior in a regime where we expect there to be a hydrodynamically driven increase in the feeding rate. So this made us really excited that maybe there's some sort of hydrodynamic benefit to this behavioral variation. So we have a couple of hypotheses for what might be happening here when these animals blink that might be leading to an increase in their feeding rate. One thing that we observe is that during a blink, particles that otherwise would pass by the ciliary band appear to take these right turns back towards the ciliary band, making us wonder whether this basically represents something like a mixing mechanism. Basically, it's very hard to mix fluids at low Reynolds number, and so having this rhythmic time variation might be a way to compensate for circulation constraints at the length scale. So we're working on using tools from mixing theory to actually quantify the spatial distribution of mixing locations around the animal to see whether there's some sort of hydrodynamically driven mechanism here underlying this. Okay, so this is, I think, what I wanted to say about Starfish for now, and I'm actually right at my target time. So I think I want to use maybe 5 minutes. Well, okay, I'll wrap this up. Sorry, I forgot I had this slide. Okay, I won't talk about what we're doing next. I'll just say that we have all this stuff on the starfish larvae. And we want to take what we've seen in this system and try to talk about this broader question of the space of possible swimming strategies and feeding strategies at this length scale. So right now what we're trying to do is we're trying to take some of what we've learned from this system and develop better three-dimensional models of different ciliary bands in 3 dimensions. And the goal is to be able to take some of what we've seen in the Starfish system, which has sort of shown us what's possible at this length scale, and use it to build a set of models that can allow us to navigate this space and see the different strategies that might be available here. And we're borrowing tools from things like topological fluid mechanics to look for very broad sort of quantitative distinctions we can make independent of specificities in the geometry. And our goal is to be able to take some of what we've learned and answer some of these larger questions about how to navigate this space of different evolutionary forces. Okay, now I'll wrap up talking about the Starfish project. Okay, so I'll skip some slides here. Okay, so I want to talk a little bit with my last few minutes about some of the people who made it possible for me to be here today. And you know, this is probably the slide I was practiced the most because I was so nervous about it. Okay, so first of all, I want to talk about someone who literally made it possible for us to be here today. As some of you guys know, one of our committee members had to bow out at the last minute on Sunday night due to an emergency. And so we were, so Manu and I were really panicking on Sunday and Monday. Those of you who saw me Monday morning probably recognize that. So I really want to thank Professor Greenleaf for coming here. He had other things he needed to do, and he was very generous in rearranging his schedule to come here. So huge shout out to him, because we literally wouldn't have been able to have this if it weren't for him. So thank you to Professor Greenleaf.
It's all thanks to me, that's right.
Critical piece in the car.
1 hour here. But it's that exact right hour. That's right, you gotta be there at the right time, right place.
I also want to thank Professor Spackowitz for volunteering to be on this committee. I think that you know, when the department sends out this abstract talking about how baby starfish swim, it doesn't necessarily get a lot of takers. So I really appreciate you volunteering to come here and listen to this whole talk. Yeah. Okay. Try to remember the order.
Your advisor.
Yeah, sure. Okay, so you know, those of you who've worked with me know that you know, sometimes when I'm working on projects, I sort of, you know, hide in the corner on my laptop and work on things. And sometimes I don't always, you know, think about the broader picture or necessarily like know about what's going on in the field beyond this. And so on both of these mixing projects, I had some really nice conversations with Professor DeBeery. And he sort of told me about some of the more like broader things happening in the field, especially on this cryptography project. He told me a lot about how people are actually using topological fluid mechanics in the field right now. And so I really appreciate his intellectual advice and also his advice on career and sort of mentorship stuff over the past year or so. Okay, so I think one thing about this slide deck is that I've talked a lot about fluid mechanics and stuff in that space, but as I mentioned at the beginning, I've spent a very large amount of time working with Mark over the years, and that unfortunately I couldn't find a non-awkward way to route that stuff into the talk. But it doesn't mean it didn't happen. It's been a major part of my time here at Stanford, has been working with Mark and learning about a totally orthogonal set of problems well outside anything I came in knowing about. So Mark has been a huge mentor to me these past couple of years. He's given me career advice. He's always been in his office and willing to walk me through even, you know, my very dumb questions about genetics. He's been willing to sort of show me how things work, sometimes repeatedly. And so I really appreciate him, you know, taking the time to just like get to know me and to try to teach me something well outside of what I came into Stanford knowing. I think I met Mark through a course I took with him, where I met with him because I had a bunch of questions about the final project. And then within 2 weeks, we were writing a paper. So I just really appreciate Mark just like wanting to work with me, his willingness to talk to this student who's interested in random stuff. Okay. So I think that I think oftentimes I think some of his postdocs get sort of the short end of the stick when it comes to credit for stuff. But I really want to acknowledge that I didn't know really any fluid mechanics when I started out at Stanford. And Vivek is a postdoc in our lab who did his whole PhD on turbulence, and he started in our lab around the same time. And he was amazing about just encouraging me to just dive in and learn about this stuff and learn about the field, and making this field that seemed very sort of like foreboding to me as someone who didn't know anything about it coming in, making it feel accessible and like something that I could do. So in addition to directly working with me on all this Starfish stuff I talked about, Vivek has also just always been there to encourage me, to tell me what's going on in the field, and to just help me sort of get up to speed and get excited about stuff. Vivek has given me a ton of just mentorship over the years, and so I really appreciate all the help he's given me. And so Vivek is actually starting his own lab next year at the University of Miami, and he'll be studying a lot of questions at the intersection of marine biology and fluid mechanics. So if the Starfish stuff seemed interesting, there will be a lot more coming from his lab next year. Okay, Manu. Okay, so I think my first like long-term interaction with Manu was when I rotated in his lab in October 2014. And one of the first interactions I had with him was he invited me to come with him to the Marine Station, where he was asked to give one of these talks to, I think, donors and like patrons of the Marine Station. So it's like this somewhat fancy event type thing where like all these like well-dressed people were there at the Marine Station, sort of mixing. And they set up this little table for Manu, and he was giving this talk to these patrons. And he was talking about Foldscope, which is this portable microscope that was invented in the lab by him and some students. And so he was talking about Foldscope and some of the outreach projects that he'd been doing with Foldscope, et cetera, et cetera. And he had this table and he had all these donors around it. And so he thought he should demonstrate the Foldscope, he should show them what it could do. And so he remembered that a few days earlier he'd found a spider egg casing in the bushes by his office. So he's like, perfect, right? They can like look inside and see like little developing spider embryos, et cetera, et cetera. So he reaches in his pocket and he pulls out the spider egg casing, and he drops it on the table. And as you can imagine, it bursts open, and like 200 like little tiny baby spiders are just like all over the table. And so all these really well-dressed people just like recoil. It's like a wave traveling across the room. And so like Manu, he sort of is curious why everyone's like panicking. He looks down and his hands are covered in baby spiders. And he's just like, this is incredible. He takes the baby spiders and he starts trying to trap them with Scotch tape, so he can make these like little Foldscope slides, and the people can look at their little legs and everything under the Foldscope. So yeah, I think it's since then it's been an incredible adventure being in Manu's lab. I think that like just his willingness to study absolutely anything and everything, and to sort of let us explore what we're excited about, regardless of whether it falls under a specific funding objective or whatever. It's just been an incredible opportunity. I think Manu has been a great mentor in that he's sort of has allowed me to work on sort of official things, unofficial things, and everything in between. I've never felt like I've been like intellectually limited to only work on what's allowed or what I'm supposed to know about already. I've always felt like if I have a question, I can take it to him and study anything that seems interesting. And I think that's a real testament to the fact that Manu's done this incredible job creating this lab where people are excited about what they work on. The people in Manu's lab are incredibly diverse in their interests and their approaches to science. And I think this really shows that Manu's mentorship style sort of transcends a specific set of topics or approaches, and just more generally is about curiosity itself and taking this really rigorous and interesting approach to problems. So I'm sure that next year, when I'm shivering in Boston, I'm going to look back on my time in Manu's lab and really get nostalgic, because I just think this has been an incredible experience that I'm not sure I'll ever be able to replicate. And so huge thank you to Manu for making really everything I just described possible. Okay, so with that, I'll go ahead and wrap up, and I'll defer back to the committee for how they want to do questions and everything. Thank you.
from the audience, and then we'll have a closed door session. So, questions from the non-committee members. Yeah, go ahead.
I was wondering if you thought about the other direction of correspondence between cryptography and fluid mechanics. And can you get hash functions and get interesting dynamical systems from those functions?
I see. So there is I think that is actually probably a better studied area. Like there's a lot of people who define stuff like permutation Lyapunov exponents for hash functions.
Oh, I see.
In fact, some of the metrics I used in the paper to sort of analyze correlations between messages and hashes were borrowed from the dynamical systems of cryptography community. I think with this paper, I was really just trying to make a point about fluids more so, because I think that the people working on hash functions have definitely come up with a lot of interesting ways to use dynamical systems.
Any other questions?
You talked about the cost of producing vortices at lower Reynolds number.
Right.
Is that all captured in, well, so in terms of increased feeding rate with increased vorticity, but there's also a trade-off with energy? Is that?
Yeah, so we were careful about energetic arguments, just because it's sort of, with an animal system, it's hard to know where all the energy is going. So our trade-off is more just in how you allocate cilia. It is true that adding these higher order modes and seeing more vortices would be more energetically expensive. We just thought it was hard to balance that against like the energy yield of feeding, because we don't necessarily know what the energy yield would be in the ocean. But that is something we thought about, is there would be some energetic trade-off there. It's just whether it's substantial relative to the animal's overall energy flux.
Yeah, kind of related to that. Or did you look at like feeding patterns and like if you do starvation and how the kind of the feeding versus swimming trade-off changes?
Yeah, so this is one thing especially with the behavior stuff, we really looked into, which is whether we could induce behavioral changes by starving. So the story isn't super clean, which is why I don't have charts on it. Like sometimes they clearly try to just like always be in this like slow swimming, high feeding mode, but it's not uniformly the case. And so we think there's maybe a lot more going on regarding how they're going to different modes. Because it also could just be that like when we starve them, they're getting tired. So it's hard to know whether it's actually strategic change versus other things.
Yeah, it's almost a multi-ambiguous sort of problem, right? Like,
Yeah.
Exploration versus exploitation.
Any other last question?